Hello, this is Dr. E, and we're going to be sharing 8.12 and 8.13 together, which is LD50 and dose response curve. The learning objectives is 1, to define lethal dose, where 50% of the organisms die, and also evaluate what is a dose response curves. Both of those are intertwined with each other. Dose response studies in LD50. Studies that expose an organism to different doses and concentrations of a chemical to measure the response of the organism. And the independent is the concentration of the chemical added to the food, water, or air. The dependent variable will be the response measured in the organism, usually death or impairment. The LD50 refers to the dose or concentration of the chemical that kills 50% of the population being studied. That helps scientists, entomologists, all those kind of people, like what just happened with the COVID-19 testing, to figure out what is the optimal dose, or what is the dose that will kill you, and therefore what won't kill you. LD50 data are usually expressed as mass in grams or milligrams per body mass in kilograms, parts per million or parts per billion, mass per volume in water of blood. So the dose response curve. Data from a dose response study, graphed with percent mortality or the effect of on the y-axis and dose concentration of chemical axis. The lowest death right here, where an effect starts to occur, is called the threshold or toxicity. So what happens is different concentrations of the chemical, concentration of the toxic in milligrams to kilogram, are added, 25 all the way up to 175. And then the number of organisms that die are counted. And that is how it's plotted, where the percent mortality is counted versus the dosage. The dose response curve are usually S-shaped, where low mortality rates, and then all of a sudden there's a huge increase in mortality as the dose increases. And it levels off after 100% mortality because no more can die. Effective dose at 50 refers to the dose concentration of a toxin or chemical that causes a non-lethal effect, such as infertility, paralysis, or cancer in 50% of the population being tested. For example, the concentration of atrazine in the water that causes 50% of the frogs to become infertile. It's the same general S shape as the LD50 dose response curve, but at a lower dose. So the toxic dose is right here, the effective dose, or the effective, even farther down, the effective dose where maybe it gets rid of a headache. The dose response studies for toxic chemicals are not done on humans. The data is collected usually on other animals such as mice and rats, and then which are used to simulate human toxicity. Sometimes they are taken to other organisms, such as pigs for heart studies, or chimpanzees or monkeys for other human studies. To determine the maximum allowable levels of chemicals for humans, we generally derive the LD50 or effective dose 50 by 1,000 to make sure that we're, the concentration by 1,000 to be cautious. Acute versus chronic studies. Most dose response studies are considered acute, since they only measure effects over a short period of time. They're also isolated to a lab, so they don't measure ecological effects of organisms dying. Chronic studies are longer term and often follow developmental impacts. For example, my mom had polio. Now, she survived polio, but 50 years later, she had problems with muscles that had not been damaged by the polio. For example, here, a study of fish from hatchlings to adult to study these sexual maturation effects of the drug. So, the practice FRQ. Here we have drug discrimination and lethality in rats. RTI, which is basically which group they were, ED50, LD50, TI, which is LD50 divided by ED50, maximum lethal. Above are data from the results of a dose response study for polonium toxicity in rats. A, can you explain how these data could be used to determine a maximum allowable level for humans? And B, can you identify the lowest dose that triggered a response in RTI number 336 rats and the dose that resulted in 50% of the RTI 336 rats dying? Thank you, and I hope that was helpful.